Okay, everybody, welcome back. We're now going to finish the chapter on microbial metabolism. This should be a little bit shorter, but we will finish microbial metabolism here. Okay, now last time we left off, we were talking about aerobic cellular respiration. So using oxygen to break down glucose to make ATP. Now, many microbes don't have to use oxygen. If we don't use oxygen, we can still break down glucose to make ATP using what's called anaerobic respiration, or a second way is fermentation. Now, anaerobic respiration, the metabolic pathways are similar to what we talked about with aerobic cell respiration. So similar to those used in the presence of oxygen. However, the big difference is that the final electron acceptor in the electron transport chain is not oxygen. Remember, in cellular respiration, the last one was oxygen, and it recombined as water and so forth. In anaerobic respiration, the final electron acceptor in the electron transport system is not oxygen, it's some other molecule. Examples including nitrate, sulfate, carbonate, and then these are the products you get. So not just water like we do in aerobic cellular respiration, but these other products. Now, anaerobic respiration it yields less energy than aerobic respiration because only part of Krebs cycle operates without oxygen. That being said, anaerobic respiration can be fairly efficient. It can provide an additional 2 to 36 molecules of ATP, depending on a whole host of factors. Now, still other microbes might not use anaerobic respiration. They might not use aerobic cellular respiration. They might use fermentation. Fermentation is the incomplete breakdown of glucose. It's also an anaerobic process. It's going to occur in the absence of oxygen, and it occurs in the cytoplasm of cells that use it. Sugar, glucose, is going to still undergo glycolysis, just like what we talked about under aerobic cellular respiration. So we're going to break down our glucose into pyruvic acid. Now, instead of the pyruvic acid getting picked up by coenzyme A now and going into Krebs cycle, all we're going to do is we're going to directly convert that pyruvic acid into some other organic compound, which is then released as a waste, organic carbon containing. So my pyruvic acid is going to be converted into some other organic compound. The compound that we produce depends on a number of factors, including the type of fermentation, the microbe doing it, and how long it ferments. That being said, the two most common compounds that are produced that pyruvic acid is converted into are ethyl alcohol and lactate or lactic acid. Ethyl alcohol is not rubbing alcohol, that's isopropyl. Ethyl alcohol would be like alcohol that you drink or cook with. Lactate or lactic acid, where might we find that? Okay, lactic acid, lactate can build up in muscle cells, especially in athletes or people working out really hard, or you just, running a marathon, working out hard. You might have heard the phrase that an athlete or a runner might hit the wall. They just get to a point where they cannot go further. You feel your muscles, particularly your leg muscles, just giving out. This is because you're working those cells so hard that you can't get enough oxygen to them quick enough to undergo cellular respiration. So instead, those cells have to switch to fermentation, an anaerobic method. The waste product of that fermentation, the pyruvic acid in your cells, is converted to lactic acid, which can build up and in extreme cases could lead to death. It's called lactic acidosis. okay? We're also going to release CO2 as a waste. We can get an additional 2 or so ATP through substrate level phosphorylation. So fermentation, everyone, is not something you want happening in your cells. In fact, most living things on Earth would not want fermentation happening in their cells. But in some of these microbes, that's what they do. Where is fermentation really used? Industry. Yeast fermentation and bacterial fermentation, used to make all sorts of products as we see here. Okay? Wines, vinegar, baked goods. Okay? Think of all different alcohol beverages. Things like that. That's all based on fermentation. All right? So we can see here our glucose is converted to pyruvic acid. That's glycolysis. Now this pyruvic acid can become a number of different waste products. We can make propionic acid, lactic acid, ethanol or ethyl alcohol, acetone or isopropanol. These are bacteria that do that, different species, and what you could use to make from them. So there are a lot of industrial uses to fermentation. All right? So here we see fermentation again, lactic acid or alcohol, or here, remember, ethyl alcohol, that's going to be used in, say, wine or beer or other products that make use of alcohol. The CO2 that's given off can be used to cause bread to rise and baked goods, and there's that lactate or lactic acid building up in muscle cells as a toxin. So if we look back at this diagram again, before we went to this side, to the left in the green, and we had our glucose being converted to pyruvic acid, and then follow this path, in fermentation, we're still going to do glycolysis. We're going to make our pyruvic acid. We have some temporary energy here, real energy here. That's through the substrate level phosphorylation. And then all we are going to do is we are going to convert our pyruvic acid into some other compound, which we talked about. So this little chart here shows us a comparison of aerobic respiration, anaerobic respiration, and how much ATP we could make. Okay, down here. All right, so aerobic, anaerobic, fermentation, there's oxygen required, what type of phosphorylation, the final electron acceptor, and how much potential ATP we can make. Okay? All right, everyone, that's a short little class this time where we just finished up microbial metabolism. Our next topic will be to delve a little bit deeper into what microbes do, and I'm going to talk about microbial genetics. That's our next topic. So until next time, have a good rest of the day, and we will see each other soon. Okay, bye-bye.